Well, I'm gonna write it down.
Our last two presentations utilize the model organism C. elegans. Here's our first group talking about antihistamines.
Hi everyone, my name is Arabella. I'm Natalie.
I'm Rob.
And our research is the effects of antihistamines on C. elegans. So our research is based on previous studies that have found a connection between dementia and antihistamines such as Benadryl. Just for some brief background, dementia is a medical condition that includes symptoms such as forgetfulness and memory loss, and antihistamines are medications used to treat allergy symptoms. So one study was done at the University of Washington School of Pharmacy, and we tracked the health of 3,500 individuals aged 65 years and older. And after a period of 7 years, they found that those who took antihistamines were more likely to have developed dementia. So while there has been a connection found between dementia and Benadryl, not enough research has been done to know exactly what that specific connection is. So this brings us to our research question. What are the effects of short-term and long-term antihistamine use on memory?
Alright, first we need to learn about what histamines are. Histamines are neurotransmitters that are responsible for wakefulness. They're also produced by the body's immune system to cause an allergic reaction. This is histamine. Antihistamines are chemicals that are produced by our body. They reduce the allergy symptoms, and this is a basic diagram. There are three classes of antihistamines. First class antihistamines are the oldest, and they actively cross the blood-brain barrier. When they cross the blood-brain barrier, it causes symptoms like drowsiness and dry mouth and dizziness. Example of a first class antihistamine is diphenhydramine, which is the active ingredient in Benadryl. That's the blood-brain barrier. It just helps keep the stuff inside the brain inside and outside the brain outside, and it selectively lets stuff go through. We chose to use diphenhydramine as our antihistamine because it's active ingredient in a lot of antihistamines and sleeping medication, so it's very commonly used. And it is a first-class antihistamine, so we know it crosses the blood-brain barrier.
So for our research, we're using these organisms called C. elegans. These are an organism that has been used for various contexts by scientists because of their characteristics. They're very small, they have a short, rapid life cycle, about 3 days to develop from an embryo to an adult. They're easy to propagate, they're transparent, and they have a well-annotated compact genome that's been entirely mapped out in sequence, and it's very similar to humans. So what exactly is a C. elegans? A C. elegans is a free-living soil nematode, also known as a roundworm. They live in many parts of the world, and they feed mostly on microbes like E. coli. They're hermaphroditic, which means they have both female and male reproductive organs, so they can self-reproduce, and they're also 1 millimeter long. So they're really tiny. If you see here, this is a plate full of worms, and then we zoomed it in a little bit. The fully grown adult is barely visible to the naked eye. So their development, their embryonic stage lasts about 14 hours, and then it develops into 4 different larval stages, L1 through 4. Each lasts about 8 to 12 hours, and then it develops into a full-grown adult. So to maintain them, we grow them on agar plates, and they have a lot of bacteria, E. coli, for food, and we keep them in a humid environment at room temperature around 20 to 22 degrees Celsius.
Okay, so C. elegans live in soil in the wild, and they sense their predator through vibrations. So because of this, they have a response to vibrations to swim very rapidly backwards, and this is separate from their normal movements, which are very slow and moving forward, although sometimes they do move backward as well. So how does this connect to dementia? Because I don't often associate birds with humans. But what we do is we use this response that they have to swim rapidly backwards, and we can monitor their effects on learning and their ability to remember using a process called habituation. So what is habituation? Habituation is defined as a decreased behavioral reaction due to repeated stimulation. This is usually involving one stimulus, and it is a non-associative form of learning. So an example of this would be if you teach a dog to sit by giving it a treat, it would associate sitting with that reward, and that's why they continue to do it. But with habituation specifically, there's no reason that they keep doing it, it's just occurring. So this form of learning is separate from both motor fatigue and sensory adaptation, which means they're still getting continuous input from the stimuli, but they're choosing to ignore it because they perceive it to be insignificant. So an example of this would be when you walk into a really smelly room, and after some time you don't notice it anymore because you're being habituated to that smell. You don't notice it because it's not a significant factor to your day-to-day life. So what are we going to be doing with this information? So when we can ensure a consistent protocol that the worms will be habituated every time, we could administer diphenhydramine at different doses and for different periods of time. And then we know that if there are any effects on the habituation or changes to it, we know that it's because of diphenhydramine and not something else.
The worm shaker was created because we needed a way to consistently vibrate the worms for habituation. Before we used toothbrushes, but these were inconsistent. The vibration motor can provide a stable and consistent level of vibration across all of the models of worm shaker. The worm shaker group previously used electric toothbrushes, but these were inconsistent due to human error. A Dickies worm shaker was used, but you can't record videos of all the voices at the same time. The Raspberry Pi version worm shaker V1, developed by me, was used, but was fragile, messy, and inconsistent. The Headstrong Shake Shaker is used on all of the currently used shakers. It has a modular base that can be put on a microscope for easy observation. The currently used worm shakers are the Raspberry Pi Worm Shaker V2, which is controlled by a touchscreen or a mouse. This worm shaker is easily customizable. There is the V2 worm shaker, which is this one, which is manually controlled by physical buttons. The Python shaker, this one, is controlled by a computer, and it is automatic. Touchscreen shaker, controlled by a touchscreen, is also automatic. The physical buttons worm shaker, this one is controlled automatically by physical buttons.
Okay, so how do we habituate C. elegans using these devices? So C. elegans can endure long-term habituation that lasts for 24 hours or more. This is important because we can administer diphenhydramine for longer periods of time and know that it's going to have an effect on their long-term memory instead of short-term. So to achieve this, we need to have longer intervals between vibrations, and you can't overload them with stimuli. So in our case, that means taking breaks from vibrations, as well as we can't just vibrate them for an hour straight because they would not be learning. So to do this, we need a way to transfer the worms onto our experimental plates, and we do that through picking. So here's a pick here. It's made of glass. Oops. So it's made of glass, and then there's a little titanium wire there. It's about that big. Platinum wire, sorry. So this is good for taking a specific amount of worms that you want. So in our case, we needed 20 worms per plate, and it's also good because you can take a specific stage of worm. So at the beginning, we talked about the larval stages, L1 through 4, and we wanted L3 worms because then when we do data analysis 24 hours later, they would be grown into full adults that we can use for processing. So we're going to be performing a few different studies, which we'll talk about later, but there are some consistencies between them. So we will pick 20 worms onto each plate, habituate them unless they're a control group, and then 24 hours collect data. And then this data will be analyzed using TRPC and then a student's t-test for significance. So based on all that information, this is the protocol which we use. So the plates are vibrated for 10 seconds, followed by a 60-second rest, and this is repeated 10 times. following that is a 1 hour rest, and this occurs 3 times total, so 3 blocks of vibration with 2 more rests in between.
For data collection, we would record on our phones and using an adapter that will allow us to connect our phones to a microscope. All our raw videos would look like something like this. They are 10 seconds of just observing the worms and then followed by 10 seconds of vibration. After the raw videos are uploaded into a worm tracking software, we are able to get data from each of the worms. For our worm tracking software, we used TRC. TRC is a worm tracking program made by MRC Laboratories, and we used it mainly to track the speed of the worms, and this allows for us to get precise and unbiased data. Using TRC, you need to make sure that there are a lot of worms on the plate, so because it will sometimes not recognize some obvious worms, and to make sure that there are enough worms to get at least a couple. So when we upload raw videos into TRC, we have to play with some parameters, and once we play with the parameters and highlight only the worms instead of the agar spots, we can save the parameters and it will save as a masked file. After they process the after TRC processes the masked files, it makes boxes around the worms and gives us data about them. Sometimes it doesn't recognize even the obvious worms, as I said, and sometimes it recognizes food spots or food trails as worms. After we find a worm with some good data, we click plot, and then it will give us a graph. If we click export to write to CSV, it will give us the data in a data sheet in Excel. Some complications we face with TRC is that it doesn't recognize some very obvious worms, as I said. And if there's a lot of worms moving on the plate and it has to recognize them, it crashes the computer. It identifies the food lines in the agar as worms. And when we started using the software, the instructions were not very intuitive, and it caused it was really difficult for them to use the software.
So we ran a total of 3 different experiments. The goal of our first experiment was to simply verify the habituation for our worms. The goal of our second experiment was to give the worms diphenhydramine and see whether that affected the habituation process. And the goal of our last experiment was to compare short-term and long-term diphenhydramine exposure. So for our first experiment, we started by picking our worms. This consisted of 2 different groups, 9 plates each group, 20 worms each. Half of them would be habituated, half of them would be our control group. So directly after picking with your habituation protocol, this, as Natalie said, was 10 seconds of vibration followed by 60 seconds of rest, 10 times, followed by 1 hour of rest, and then 2 more times. And then we would wait 24 hours before collecting our data, and then we would take a video of each plate, 10 seconds normally, followed by 10 seconds of vibration. So these are our expected results. This first protocol has been run for several years now, and this is what we usually get. For the non-habituated worms, because they aren't used to the vibrations, they would have a certain speed before vibration, and then during vibration, they would show a significantly higher speed. For the habituated worms, because they are used to the vibrations, they wouldn't show any significant difference.
Okay, so these are the results that we got from this experiment. So our non-habituated group and our habituated group, for some reason, did not show statistical difference. And as you can see with the error bars, they are overlapping. And because of this, we can't confirm that they're habituated. So we've run this many times in the past. This is actually one of the first times this has happened. But because of this and the experiments later on, so because the later experiments were testing diphenhydrate's effect on habituation, we can't use that data because we can't confirm if habituation was actually working. So some factors that could have gone into this was our N value. So we had 9 plates, each with 20 worms, so that we had a lot of data to work with. But partially due to TRC, it trashed about half of our data. So we didn't have a lot to work with. It could have also been the numerous shakers we were using, because some of them we hadn't used before, and and maybe they were inconsistent, but we're really not sure this time. But we will still walk you through experiments.
So experiment 2 follows pretty much the same protocol as experiment 1, except this time we're seeing if diphenhydramine affects the habituation process. So this time we have 4 groups of worms, each with 9 plates, 20 worms each. And then this time we're giving 1.3 milligrams per milliliter of diphenhydramine to half of the worms, and we're exposing them to the drug for about 24 hours. And then directly after picking is when we run our habituation protocol for our habituated group, and then 24 hours later we collect the data. And then our third experiment was to see whether short-term or long-term diphenhydramine exposure would affect the habituation process. So this time we had 6 different groups of worms. We had our control worm with no diphenhydramine. We had our 2-hour exposure to the drug, and then our 48-hour exposure. And then everything else follows the same protocol with habituation and data collection. Okay, so for the future, we have a couple areas for improvement. Of course, we have to determine whether habituation actually works. It should, but there are definitely some sources of error. We would want to run the experiment multiple times to verify our results. And then because we have so many shakers, we want to see which is the most effective, and there are a lot of differences between the different shakers. We want to give a special thanks to Dr. Jessie Mass, Abby Gerhardt, and the rest of the scientists at the New Hampshire Academy of Science and Near Peer Mentors, and also to our Academic Associates for having us here today. We have a couple of names to show you. Oh, these are references. We have C. elegans. We have worm, the roundworm cells, free living, malfredid, nematode, I can't pronounce it, elegans. And then before we take questions, we have a question for you. We're skipping that one. If C. elegans were pants, could wear the lip pants. You can think about that. Alright, let's take questions.
Good question. One, what was your favorite part about your research? Had a great presentation. And then, is it possible with the way that your data is organized that you could answer your shaker efficacy question without extra experimentation?
I don't think so, because there are also a lot of other factors that could have gone into it, and we can't really verify if it happened because of like diphenhydramine, other factors in the environment, or if it was really the shaker.
Okay.
So I think we would have to do individual tests on every shaker and see if
So you don't have like.
Well, another thing is that we would have like the same plates on the shaker, so that like if there was a difference, it wouldn't really. Like,
So say if we had
mix
like
things
9
up.
plates, we would have 3 on 3 different shakers.
Okay.
So it might be tough to
How do you guys plan to, how would you guys plan to like try and figure out what went wrong?
I think we would probably just like eliminate factors. Maybe like if we have a shaker that we know has worked in the past, try to do exactly what we did this time, just with that shaker, and just do that with all the factors that it could be. And maybe play with TRPC and see if there's a way that we can get that to work better and really see the worms.
Yeah.
Why did you guys choose to use TRPC? It seems like you had a lot of problems with it.
So the alternative is to sit by hand and plot out the points of every single worm, and then you have to do the math and figure out their speeds, which sounds quite honestly miserable. And it also introduces a lot of bias, and then there is a software that is more efficient, but it is incredibly expensive and not quite realistic to use.
So if we go like the same back to the beginning here, when you talked about the risk to you, could you let everybody know, because I think some people might not have appreciated why you're doing this and why it's so important. Can you
Yeah, so-
say that, and then I got another question.
Like as I mentioned before, there's been previous studies that have researched the link between dementia and using antihistamines like Benadryl, and so since Benadryl is so commonly used, that's why we decided to test that one out. And yeah,
Because diphenhydramine isn't just in Benadryl, but it's also in like NyQuil and things like that. So a lot of people are using it every day if they realize or not. So we were really wanting to see what the effects of this can have because people really aren't quite sure.
So, and then the other one is kind of a logic question. If you got a crappy program that you have to use 20 worms on, and is really laborious and generates a lot of problems, why don't you just go back to the easy way of using maybe 3 worms that you can track kind of manually, and then at least you get a reasonable end sample that is reliable. And then lastly, what advice can you give us for the next session that might be trying to repeat this? Would you tell them to maybe go back to the other one, or continue with Chertsey Health, or what?
So I think for the second group of research, we were planning on doing other diphenhydramine experiments. So I think we would mainly just have them like focus on this one again because it didn't go as we planned.
and for your question about why 20 worms, a lot of the worms would die on us.
And it doesn't take super long to pick the 20 worms. Poor Raj had to do most of it.
He's really
But I think it's more efficient to pick more worms than it is to sit and track all of them, and it introduces so much bias that it's just not worth it.
I'm curious about the experimental design about using habituation to speak to human memory, and I'm wondering if C. elegans has been used as a tool to assess human memory in other experiments.
Yeah, they're kind of the main, I don't know about the main, but they're really, really commonly used for human research because of their like similar genomes and other factors like that. And because of the comparison to like their ability to learn and like remember habituation, we can see